Take a minute and set up your paper. We're going to do the second half of B4A, and it's compare and contrast scientific explanations for cellular complexity. Pause this and write this stuff down, and then you can start it back up and catch up with the rest of us here. So when we talk about comparing and contrasting cellular complexity, again, it seems like there's going to be more than one theory we're talking about, but it's not, because it's explanations for cellular complexity. God, they're loud. I have no idea. They've got someone out there taking pictures, probably. Okay, so we're going to do this in two ways, two pieces. We're going to talk about this. We're going to talk about, first of all, the genesis of cells and cell complexity. Okay, so when I say genesis, what does that mean? Genesis means what? Genesis means the creation of, right? How did they start? The origination of them. So we're looking at the genesis. How did cells begin, right? I mean, we talked about evolution. We said, well, here is a living thing, and here's how it evolves. But how did the first living thing come about? So that's what they're talking about here. And so this is a little bit weird because it's almost counter to what we've talked about before. Remember we talked about spontaneous generation, yes? And spontaneous generation says, oh yeah, mice come from old dirty shirts, or maggots come from rotten meat. So basically life can come from non-living things. And we figured out that wasn't true, correct? But now we're going to talk about something, and it's basically, they've given it a different name. It's called abiogenesis, but it's still basically spontaneous generation. So it seems a little bit weird. So the number one theory of how life began is a theory called abiogenesis. It's all one word. And abiogenesis is talking about how did life form. How did life form? If we look at that word abiogenesis, if you stick, okay, it's Latin. If you can say the word abiogenesis, you're speaking Latin. So A on the front of a word means no or not. What's bio mean? Life. And genesis means creation, right? So if we translate that directly from the Latin, it means no life creation. Or life came from non-living matter. Yeah, so we basically, by another name, spontaneous generation is what it's saying. So it's a little bit weird, but that tells you that they're not quite sure what's going on, doesn't it? When you get something where you're thinking, hey, these two things are counter to each other, then it probably means that they don't have a good theory, they don't have any good evidence, and so they're saying, hey, we're not sure, we're putting this out there, but that's about as far as we go. Okay? So how did life begin? What's cool is that they have some evidence for different processes, but they can't put it doesn't fit all together just yet. So there's one experiment that'll be in every single biology book that's written. And this is a famous experiment called the Miller-Urey experiment, which is really kind of cool. What they did is they recreated conditions on Earth from 3.5 billion years ago in the lab. Okay, BYA. So they recreated conditions on Earth from 3.5 BYA, billion years ago. That's a standard abbreviation. If it were millions of years ago, it'd be MYA, right? So they recreated these conditions in the lab, in a flask, like a beaker. Okay, so let me explain that. You know the Earth, it was formed about 4 billion years ago, right? Life didn't start until 3.5 billion years ago. And they have fossils. Bacteria can form fossils. So they have bacteria fossils. If you drill down into rock, you can drill down and you find evidence of life. You find evidence of different chemicals. What was the planet like during that time? So the farther down you go, the farther back in history you can get. You can tell what types of chemicals were present. You can tell, in many cases, a geologist can tell what type of temperature was in the atmosphere, whatever things were going on there. So they can tell a lot. Geologists can tell a lot about what was the atmosphere like so many billions and millions of years ago. Okay? So what these guys did is they used information from geologists, and they figured out, well, life started in the oceans, so what was our ocean like 3.5 billion years ago? What was it like? And they made that in a beaker. Okay? All the chemicals that were in there, they even got the right temperature, and then what they did is they ran some electricity through it, like lightning, yeah? And they saw what happened. So they made this material that had all the chemicals that they thought were present there, and then they shocked it, like lightning strike or something. And then they checked all the chemicals that were in there, and they found that there were new chemicals in there. Specifically, there were amino acids. Why are amino acids so important? Yeah, amino acids make proteins. You get a chain of amino acids together, you call it a protein. That's hugely important because you are made of proteins. All your body parts are made of proteins, basically. You have a little bit of fat, you have a little bit of sugars in you, but the main bulk of it is protein. So that's really important. So the first thing is that they were able to make amino acids. They didn't force it to happen. All they did is they ran electricity through it and then looked at stuff afterwards, and they found out, holy crap, there are amino acids in here now. The second thing is that they created organic molecules. Do you need someone to help you carry your stuff? You need someone to help you carry your stuff? Okay, so pick someone and help them carry your bag and stuff. Okay. All right, so they were able to make amino acids, and also what they did is they were able to create organic molecules. And I'm going to put the word complex right here. Complex organic molecules from simple inorganic molecules. And I'm going to put two other things in here, and it was in the primordial, okay, this is a weird one, primordial soup. Have any of you guys ever heard that phrase before? Primordial soup. Primordial, like, you know, before life was around, like really ancient, early times before life. So when they talk about what the oceans were like 3.5 billion years ago, it wasn't like the oceans today. It wasn't just water with some stuff in it. It was like a soup of all sorts of chemicals and stuff inside there. It was crazy. If we tried to get in it, it would kill us. So it was nasty, nasty chemicals. The atmosphere, if we time warp back there, it would be like, you know, death to you, because you couldn't even breathe the atmosphere out there. It was nasty, nasty, nasty situation. But it was perfect for making chemicals. I mean, if we make chemicals in the lab, we try and make stuff in the lab, you really have to have fume hoods and all sorts of stuff, because when you're making complex organic chemicals, you use some nasty stuff to do that. And so my AP chemistry class, it's inorganic, because we don't have the setup to deal with organic stuff. Organic chemicals are always a lot nastier than the inorganic stuff is. So they created complex organic molecules from simple organic molecules that they made that they had in their primordial soup. So they didn't force this to happen. Once they had the chemicals there all together, all they did was run electricity through it and then looked at it afterwards, and they found, oh my gosh, look at all this crap there. So now we're going to make a little short timeline here. So they started with simple molecules. Oh, yeah, there you go. They started with simple molecules, and then they were able to make more complex molecules. And then what they did is they found out if once they had these more complex molecules, they actually even started making more reactions, carrying out more reactions on their own. They're called metabolic pathways. Okay, you know that big poster I showed you yesterday, or whenever, day before, whatever, with all the, it looked like a big map? Those were all the metabolic pathways, the chemical reactions that were going on in the body. So those are the metabolic pathways we're talking about. A lot of times it's like when you hook amino acids together, what is it you form when you hook a bunch of amino acids together? Yeah, the formation of a protein is a metabolic pathway. Cellular respiration is a metabolic pathway. Photosynthesis is a metabolic pathway. So I'm going to put respiration and photosynthesis here because those are two that you know already. Here's the difficulty here. Once we had some of these standard metabolic pathways, how does it go from that to a cell? And I'm going to put a big question mark here, and I'm going to go on to living cell. So we have evidence from going from simple molecules to more complex molecules. We have physical evidence going from more complex molecules to metabolic pathways, but then there's this big jump that we don't know how to go from the metabolic pathways to get to a living cell. There's no evidence. There's nothing we can look at and say, yeah, this is how it happens. Yes? So that's why they're coming up and saying, well, maybe it's abiogenesis. The life is going to happen from the non-living things, the chemicals that are here. Yeah? So there's a gap. We don't know. How did it get from here it is, a bunch of chemicals non-living, to now here's a cell? That's where it's gap. That's what's happening here. So this area right here is still under research. It's still being researched. Don't know. Now again, if you're a Christian, you think God did this, and you could say easily, well then, bam, there's your miracle, the miracle of life right here. But again, it's not that scientists is anti, it's that science is saying, hey, if we can't see, hear, taste, touch, smell, then we can't deal with it. This is our tool, this is what its limitations are. It's like taking a microscope and trying to screw a screw in the wall with it. It's not the right tool. It doesn't work that way. So the science is a great tool, but it can't do this stuff unless can we see, hear, taste, touch, or smell, and that's it. Okay? So this is what we're talking about when we say that actual abiogenesis, the creation of the cell itself. And then the other thing that we talk about when we talk about the complexity of the cell is one that's more common. And I have not seen, even though it's on the teak, I have not seen any questions at all talking about the endosymbiotic theory. I don't know why, but it's just there are no questions. So we're not going to spend a ton of time on it. But when we talk about explanations, for cell complexity, well, the first thing you need to look at and remember is the cell theory. Do you need to write that down again, or will you go turn back a page and look at that? Yeah, it's really easy just to turn back a page and say, oh yeah, cell theory. All living things are made of cells. The cell is the basic unit of structure and function of an organism, and the cells come from pre-existing cells. Oh, do you see a problem right there? Cells come from pre-existing cells. Abiogenesis. Life came from non-living. You see a problem? Yeah, anytime we have the theory where we're missing a chunk of it, it's like, yeah, they're not matching up with what we have right here. So the cell theory is one thing when we talk about it, but the other piece that's associated with that is called the endosymbiotic theory. Another ugly Latin word. Okay, the endosymbiotic theory. This one actually makes a whole lot of sense. It's pretty glorious. So the endosymbiotic theory, you need to know what that endosymbiotic... Okay, so endo and exo are opposites of each other. Which one means outside? Exo. So endo means inside, right? Symbiosis refers to a really close relationship between two different species, yes? Okay, so endosymbiotic, it means one species was inside the other. That's what's going on. So, like right now, you have some species, other species inside of you. Like in your large intestine, you have E. coli in it. You have E. coli, and it has a nice balance of E. coli. So if you have not enough E. coli, you're going to be constipated. Yeah. If you have too much E. coli, just a little bit too much E. coli, you're going to have diarrhea. So you want a nice even balance of that. Now you can also have so much E. coli that can kill you, which you would not die nicely. It would be painful. But there's a nice balance. Maybe sometime years and hundreds of years in the future, maybe those E. coli will actually become a part of our bodies, where they're not separate entities, they become a part of us, they become a part of our cells. Maybe that's evolutionarily what's going to happen. So in this case, what they're saying is, if you look back evolutionarily, at one point you had two separate cells, and one kind of went inside the other, and the bigger one is going to be what it is going to be more protection, and the little one is going to give off something that helps the big one. So it's a mutualistic relationship there. Okay, so this is what they think happened. So one, you have prokaryotic cells, which are bacteria, remember? That lived in a symbiotic relationship. So you have prokaryotic cells that lived in a symbiotic relationship and I'm going to word this strangely, but this is the way it's typically done, with ancestral eukaryotes. Remember, we could go way back in our ancestors. We look at a common ancestor and go back farther and farther in common ancestors and stuff. So this would be the common ancestor with the eukaryotes. So it'd be an ancestral. So it's not the eukaryotes of today, but it's their ancestors.
What's the last word on behind after lived in?
A, symbiotic.
Oh yeah.
Yeah. So they were inside the ancestral eukaryotic cell. So here's my ancestral eukaryotic. Okay, so what that means is bacteria aren't all the same size, right? You can have big bacteria, you can have little bacteria. So at some point, everything started off. The first living cells were bacteria. And there were no eukaryotes, they were just all prokaryotes. And then over time, they evolved, things changed, they had different characteristics, mutations occurred. And so maybe at some point you had this cell, this bacteria, and part of its cell membrane came kind of in the inside and wrapped around the chromosomes. So it was like a nuclear membrane that wrapped around the chromosomes. And the chromosomes, with that mutation, the chromosomes were more protected, so less mutations occurred. And then that guy passed that trait on. And that was very successful, keeping those chromosomes safe. That was very successful, so they reproduced. So that could be happening here. So as you get bigger, when you have that DNA that is protected, you can get bigger and bigger cells. They can get healthier and bigger cells. So bacteria are a lot smaller than eukaryotic cells. Right, so now you have a big cell, it's got maybe a precursor, kind of sort of nucleus, and this little bitty bacteria is living really close to it, because bacteria eat other bacteria too. They eat stuff. They eat things, right? So the bacteria, they're hunters. They're little bacteria that hunt down and kill other bacteria. Yes? So these little bacteria might stay beside the big one, the big cell, because it's there for protection. So if you have single-celled organisms, they eat other stuff. Yeah. So I mean, they're like little organisms all on their own, and they go out and munch and eat all stuff. So anyway, this bacteria was able to get close to this organism and be safe. And so as time goes by, that became it became a symbiotic relationship. And it gets to the point where that little bacteria actually went inside the cell membrane, and so it stayed inside the cell membrane and was safer still. So you have that endosymbiotic thing going on here. So the prokaryotic cells that lived in a symbiotic relationship. Okay, symbiotic means you've got two different species living close together, right? Do you remember mutualism? Mutualism is when they're living close together like this. It's positive for one and positive for the other. It's both pluses. Unlike parasitism, where it's plus for one but negative for the other. Yeah? Mutualism, parasitism, commensalism. You might recognize those words at least. So they were in this symbiotic relationship. Two, they evolved so that they couldn't live without each other. Since they're both benefiting from it, they evolved so that they couldn't live without each other. And then three, that prokaryote, those little bacteria that were in there that got eaten in and lived inside the other one, those little prokaryotes became what we call organelles. So those prokaryotes eventually became organelles. Now you're thinking so far it's like, I'm not seeing how cool it is. So let me explain how cool it is. I'm talking two prokaryotes, two organelles, mitochondria and chloroplasts. I'm going to ignore chloroplasts because we don't have those, right? So mitochondria. Did you know mitochondria have their own DNA? They have their own DNA. You do not have any genes of your cells that make mitochondria at all. Mitochondria make themselves. So the mitochondria reproduce. They basically make clones of themselves. That's it. So you can't make the mitochondria. The mitochondria have to make themselves. Now, if you take the mitochondria out of the cells, they can't survive on their own. If you take the mitochondria out of the cell itself can't survive on its own. They have to be there. But they have their own DNA. Chloroplasts have their own DNA. So when you look at this stuff, you're thinking, oh my gosh, I can kind of see what they're talking about here. Mitochondria and chloroplast both have their own DNA. And ribosomes. They have their own ribosomes too. Do you see now why we think, oh, hey, those could have been organisms on their own? And they were incorporated into those cells way back historically. So again, you have no genes to make mitochondria. As a matter of fact, all of your mitochondria come from your mother. So if you look up here on this picture, here we have the sperm and we have the egg. The egg's massive compared to the sperm. So the sperm have to travel, right? They've got to swim. So they are condensed. They are streamlined, condensed down. The only thing, here's the head, the neck, the tail. The only thing in the head of the sperm is the chromosomes. That's it, the DNA. That's it. Here are where the mitochondria are. And then these are like microtubules. You don't care about that. So when the sperm, the very first sperm, touches, there's a chemical on the outside of the egg. The chemical here reacts with the chemical here, and immediately, within nanoseconds, there's a chemical barrier that pops up around the egg. Only the head of the sperm can get through. The neck and the tail drop off outside. So the mitochondria that were in here from the father never make it into the egg, the fertilized egg. It's just the DNA. Whatever mitochondria were in here, those are the ones that are going to be passed on to the fertilized egg. So the mitochondria in here all just have to reproduce, and they just clone themselves is all. So we have, from you guys, every single person in here, your mitochondria just comes through the maternal line, always through the maternal line, never through the father, because that mitochondria never made it into the egg. That mitochondria, do you guys ever watch those CSI shows? Yes, no? Yeah, have you ever heard them talk about mitochondrial DNA? If they can't get good DNA, let's say, okay, so somebody's found a dead body, a skeletal body, the remains of something out in the field somewhere. And most times if it's too old, they can't get DNA out of it. And so one of the DNA sets that's easiest to get and still can survive is the mitochondrial DNA. So sometimes they can get mitochondrial DNA. It's not as good a fingerprint as the person's DNA, but they can get relationships. So they can kind of cross people off their list by saying, yeah, this is not their mitochondrial DNA. So they can narrow it down to like a female line. So they can really narrow it down pretty well. But the mitochondrial DNA is actually a little bit tougher and can last a little bit longer.
So the sperm really isn't that important.
Yes, it is. Because that's the other half of the DNA. Yeah. It's got half the chromosomes. The egg has the other half of the chromosomes. The egg is bigger, it's got more all the other parts to the cell in it, but if you don't have those chromosomes, then you have nothing. It's got to have both of them. Yeah. So as for the other organelles and cell parts, no, it's not coming from the sperm. It's the chromosomes that are coming from the sperm. Yeah. Yeah. Okay, so when you look at this, they both have their own DNA and their own ribosomes. The next thing is they can reproduce themselves, which is weird. And then the third thing is that they both have a double layer membrane, which is another weird thing and important. Your cell membrane is a double layer membrane. All cells have a cell membrane and they're double layer membranes. Organelles, the vast majority of organelles inside your cells, they just have a single layer. It's just a simple little, kind of like a baby membrane. It's not that good. But the mitochondria and the chloroplast have a double layer membrane like all the other cells on the planet. The only other thing in your cell that has a double layer of membrane is the nucleus, which we think developed from like a pooch end of the cell membrane and it wrapped around the DNA inside there. So it's like a mutation that happened. So again, that's all you have on there. So let me show you a picture really quickly. Last time I taught AP Biology, I used this book. It's a beautiful book. And one of the things I did when I was teaching AP Biology, it's such a good book, is the first thing that we did when we looked at a chapter is said, I don't want you to read through the chapter. All I want you to do is I want you to look at the pictures, the illustrations they have in here, and just read the captions underneath because their illustrations are phenomenal in here. So here, I'm going to show you the little picture first. Here is what your double, when I say double layer of membrane, do you see the little balloon with two strings on it? There's one layer, and then upside down from that, the balloon with two strings on it. Those are lipids. Another name for lipids is fat. Yeah, so what are proteins made of? Amino acids. What's another name for lipids? Fat. Fat. Two different chemicals. We have lipids, also known as fats. We have proteins made of amino acids, yeah? So here are your double layer of lipids, and this thing is a protein. So when we look at these membranes, when we say double layer of membrane, it's got two layers of the fats, the lipids that are there, and usually they have protein chunks in them. Now this is your cell membrane, but the mitochondrial and the chloroplast membranes are made in a similar way, which is weird because typically only the whole entire cell, one organism would have that layer of membrane. So it's, again, kind of pointing to the fact that, hey, we think maybe these chloroplast and mitochondria were at one point little bacteria off on their own and they got incorporated into a cell through this symbiotic process. Yes? Okay, go ahead and take out your color sheet of the cell, and we're going to list the parts of the cell, and we're going to do the structure, brief structure, and function for each part of the cell.